Well, good morning, everybody. It's Friday at 11 a.m. That can only mean one thing. It's time for the Fralin Biomedical Research Institute Pioneers in Biomedical Research Seminar Program. Before I have the pleasure of introducing the introducer for today's event, as always, I would like to remind people of the upcoming seminar that we'll have next week, put in a little plug for it. And same time, same station. Next week, we'll have Dr. Evan Scott, professor at Northwestern Medicine, Feinberg School of Medicine. And Dr. Scott will be telling us about engineering the bio-nano interface for enhanced nano-immunotherapy. So I hope everybody will join us next week as well. So without further ado, before I introduce the introducer for Dr. Ross, I want to remind everybody to please use the Q&A to enter your questions. And then Dr. Gordy, as the host, will read the questions to our speaker during or at the end of the talk. So Rob Gordy, turn it over to you to introduce Dr. Ross today.
All right, thanks, Mike. Morning, everyone. So it's my pleasure to introduce Bob Ross from University of California, San Diego School of Medicine. Bob is currently the Assistant Vice Chancellor for Health Sciences and Academic Affairs at his institution, and also serves as a Professor of Medicine, as well as acting as a staff physician on the Veterans Affairs Hospital associated with UCSD. Bob earned his MD from the University of Southern California, Keck School of Medicine, and served as an intern in Los Angeles County USC Medical Center, did a residency in internal medicine at Cedars-Sinai. He also did training at the NIH and a cardio fellowship training at UC San Diego Health. His research is focused on heart failure and in particular the molecular basis of heart failure. He's widely published, and in fact, they had a beautiful paper just recently in Circulation Research, which I'm sure we're going to learn more about today on a protein dear to my own heart, zonular occludens 1. He serves on numerous study sections and review panels. And in fact, you know, I've long known Bob, and we've had numerous collegial and friendly interactions over the years at study sections, but also at scientific meetings and so on. Bob is also, in addition to being an outstanding researcher, he's also a passionate teacher and involved in various aspects of medical education. He is the principal investigator on an NIH T32 training grant on cardiovascular development, physiology, and disease. And he also runs a summer training program for undergraduate students who are underrepresented in the sciences and have an interest in pursuing further education in MD and PhD. And he was also the recipient of the UCSD Vice Chancellor Award for Equal Opportunity in Affirmative Action and Diversity. So, you know, Bob is the full package, outstanding researcher and passionate teacher, and also a great colleague. And it's my pleasure to turn it over to him and hear about his current research.
Thank you so much, Rob, and thanks for the kind introduction. So can everyone hear me? Am I audible?
Yes.
Okay, I'm going to share my screen and get started here. And is that working? Very good. So thanks for the invitation, Rob and Michael, and really the entire institution. I wish I was visiting in person in Virginia, but as I mentioned to Dr. Friedlander this morning, it is nice to be able to teleport to Virginia at a moment's notice here. This is the first full day Zoom talk I've given in the midst of a pandemic, in the middle of a presidential election. So this is quite an honor. So as Rob mentioned, we have a lot of interest in the molecular basis of heart failure. And what I'm going to focus on this morning, and it really does feel like bringing coals to Newcastle, are ZO proteins as I focus on my talk. What you're seeing here is, it looks like it's something that someone painted, but this is an actual photo that I took at Torrey Pines State Park, right near UCSD. I can't say it's a view from my office, but pretty close. And we have people flying around on nice days, and in San Diego, those are pretty frequent. So we've had a lot of interest over now, it's hard to believe, many decades in really understanding the molecular basis of heart failure, and really focused on the basic science of what genes are involved in taking a normal appearing heart with normal function. I'm showing here with an echocardiogram, either through a pathway where the heart hypertrophies and then ultimately dilates and is failing, or sometimes avoids this hypertrophy and moves directly, or at least as we see, towards a dilated cardiomyopathy. And as a component of many of these studies, we've also become more and more interested in heart rhythm and conduction. So to put this a bit in context, if we think about cardiomyopathy and heart failure in the US, the incidence of cardiomyopathy in the pediatric patients for children less than 18 is about 1 case per 100,000. And about 0.6 per 100,000 that have full-blown dilated cardiomyopathy. Once one has reached adult age, the heart failure diagnosis is projected, this is from the latest statistics from the American Heart Association, to balloon from our current estimate of about 5 million people to 8 million by 2030. And there's approximately 1 million new heart failure cases each year. It's now diagnosed in as frequently as 1 in 100 individuals over age 65. And the cost in the US is absolutely enormous, estimated when this was last done now a number of years back to be $31 billion. And 10 years from now, it will be as high as $70 billion of cost to the health system, or about $250 for every single person in the United States. If we think about the causes of dilated cardiomyopathy, which we're very much interested in, chronically there's, as we're learning over time, a tremendously diverse group of genes. There's those that are autosomal dominant with variable penetrance and expressivity, as we've seen in many of the diseases that we're interested in. In children, there may be as many as half of those that develop cardiomyopathy that have some form of myocarditis, and some that have neuromuscular diseases as part of this process. There are those diagnosed with cardiomyopathies that have potentially reversible causes. I see these frequently in the hospital, and these are very tough to deal with. Not so much tachycardia induced, though that always comes up in terms of, for instance, someone that has atrial fibrillation and has an echocardiogram that looks as if their heart is failing, but then once they're back in sinus rhythm and the echo is repeated at a normal heart rate, we realize that that may or may not be so. Stress-induced cardiomyopathy, alcohol's common, chemotherapy-induced, which has really come to the fore as the burgeoning subspecialty within cardiology of cardio-oncology has come to be recognized, infectious causes, and of course, pregnancy. And then for hypertrophic causes, there's been a lot of attention to the various sarcomeric protein mutations that have been identified in as many as 30% of those causes. And again, those have frequently incomplete penetrance and expressivity. So what about our advances as far as treating heart failure? The survival in heart failure, once it's diagnosed, has improved over time. But remarkably, once one is diagnosed, the 1-year mortality still is as high as 30% in those over 65. And once a heart failure patient is hospitalized, the 5-year mortality can be as high as 40%. In many of the things that we're interested in when we're thinking about mechanotransduction in the heart, and particularly the impact of something like hypertension in combination with, say, an underlying gene variant or defect, is very much interested in this concept of a double hit hypothesis. And it's felt to be that, for instance, 75% of the heart failure patients may well have some element of antecedent hypertension, which may exacerbate an underlying gene variant. So do we have any new therapies for heart failure, particularly if we think about, as we, and I'm not going to get into this variation, the distinction between heart failure with reduced ejection fraction, that are those, as I showed earlier, that have dilated cardiomyopathy, for instance, and depressed ejection fraction, or those with heart failure with preserved ejection fraction. One thing I'll point out, we have very few new therapies, but recently, and I guess we can think of this in various terms, myocardia has a new therapy which may be useful for treatment of hypertrophic cardiomyopathy. It's a unique therapy. I have no alliance to this company, but interestingly for the biomedical scientists, there was such excitement about myocardia's hypertrophic drug, they were recently acquired by Bristol Myers Squibb for $13 billion. So we'll all take pause there. So let's get to the point of today's talk. We've been studying genes on the cell surface of the cardiac myocyte now for many, many years. We've spent a long time investigating the function of some of our favorite proteins, the integrins on the cell surface, and how they transmit signals into the myocyte. And then along with that, a host of proteins that connect directly or indirectly to integrins and might control their function, including kindlins, talin, and what I'll focus on in part today, vinculin. And these proteins are at that costameric site, which couple the sarcolemma all the way through to the sarcomere. And many of those same proteins are also found at the cell-cell junctions between myocytes at the important intercalated disc that I'm sure many on the call are very much interested in, where we think about gap junctions, fascia adherens junctions, and desmosomes, and all of the proteins that we've learned over time that are components of these various junctions in the intercalated disc. And as we've learned more about them from human genetic studies, as well as studies in mouse models and cell culture, we know that many variants in these proteins can lead to various forms of cardiomyopathy. And so what I'm going to focus on for the first part of the talk is a protein we studied for many years, one called vinculin that I mentioned is also at the costamere and connects indirectly to integrins. And then I'll spend time talking about, as Rob mentioned, zonal occludens proteins and where we've gone with that and how that work is progressing. So what is vinculin? Vinculin is a 117 kilodalton, 1,000 amino acid protein. It's absolutely ubiquitously expressed, and I'll mention this in that many of the proteins that we've studied in some parts are frustrating, as I'll get to at the end, as to the question that's always asked about their relevance to disease in general, and specifically cardiovascular disease, in that being ubiquitously expressed, if there's variations in many of the proteins, and vinculin is one, sometimes the organism does not survive. It's highly conserved from worm to man, and it provides a structural link between actin and the cell membrane. Interestingly for us, and one of the things that got us excited about studying vinculin in a cardiac myocyte when we first did this many years ago, is that there is a muscle-dominant splice variant isoform of vinculin termed metavinculin that's specifically expressed in heart and in skeletal muscle. So here is a crude structural diagram of vinculin. It's a multivalent protein binding, as you see, to catenins, talin. It has a head-tail association, as I'll show you in a moment, an alpha-actinin binding site, this what's called a hinge region where these proteins are bound, vinexin, ponsin, and VASP. Multiple other alpha-actin binding sites, phospholipids, paxillin that we've also studied in the past. And this insertion that I've mentioned called metavinculin. That's just the 68 amino acid insertion site as diagrammed here. So vinculin is found in the inactive form where its tail binds to its head as shown here. And when many of these proteins then bind to vinculin, it becomes activated. I'm not going to spend more time on this conformational switch that occurs further than mentioning this. And so what interested us as we got into studying the role of vinculin, and for most of the work that we do, we utilize genetically engineered mouse models, is that vinculin was located both at the costameric site that I showed you earlier on as it binds to talins, which bind directly to the tail of integrins on the cell surface, but it's also found at the intercalated disc, as I'm diagramming here. So to investigate vinculin, we created a floxed mouse here, as I'm showing, where we floxed exon 3 in vinculin, utilizing several different Cre drivers, one that we had made in the laboratory quite some time ago that is driven by myosin light chain 2 ventricular. Ju Chen created this original Cre model, one of the first that had cardiac specificity, and one that many people around the world have used, the alpha myosin heavy chain Cre, to take out floxed alleles specifically in cardiac myocytes. And with that, we were able to derive, and this is how I'll term a cardiac-specific knockout, a cardiac-specific vinculin knockout utilizing these various drivers. Well, we expected immediate heart failure to occur with these various Cre drivers. And what we saw, which was quite interesting, is in essence that these mice had two separate phenotypes. Early on, we would see mice that looked perfectly well in the cage on one day, and we'd show up the next day and they were dead in the cage. And what we learned by doing conscious telemetry and other electrocardiographic studies is that these mice were dying with, for the most part, what looked like torsade de pointe, ventricular tachycardia that they could not recover from. But there was a subset of mice that once they escaped this period, and there was some gender variance, and I won't get into that today, that these mice ultimately, as we had predicted from the outset, given the presence of vinculin both at costomeres, which are felt to be structurally critical for stabilizing the cardiac myocyte, as well as at the intercalated disc, that these mice ultimately did go on to have significant dilated cardiomyopathy and completely die by age of 20 weeks and upwards up to 30 weeks. And when we investigated these mice at the electromyographic level, we saw here is a control littermate where the intercalated disc is well structured. And here, in essence, in the vinculin knockout, well before there's evidence of heart failure, we saw there was almost complete loss of the electrodense material at that intercalated disc. We, of course, went on, particularly since we were interested in why these mice developed the ventricular tachycardia that they did, to evaluate the connexin 43 expression. And although there was some spreading of connexin 43, this is in ventricular myocytes, there wasn't dramatic loss of connexin 43. And again, we made sure that these mice were very early on in their course. They had loss of vinculin, but no evidence of heart failure at this stage. But we see that there's preservation, but some distortion in connexin 43 in the left ventricle. So given the rhythm disturbance in these vinculin knockout, we asked what role might vinculin play in the stabilization of connexin 43 gap junctions. And this is work that was carried out by Elise Emlich-Harf in the lab now a number of years ago, and she's moved on to bigger and better things, but still at UCSD. We specifically were interested to see if vinculin and Zo1 both physically connected and were co-localized within the cardiac myocyte, and indeed that is what we saw. here by confocal, we saw that vinculin and Zo1 really co-localized quite well here within the intact tissue in the mouse left ventricle, and here even within dissociated myocytes, as we're showing. The residual Zo1 in our wild-type mice, we could show that Zo1 was dominantly expressed in the left ventricle at endothelial cell sites. Here we're using PCAM as a marker of endothelial cells. We see Zo1 clearly at intercalated disc sites, but by co-localization, we can see that there's significant expression of Zo1 at all of the sites marked by PCAM in endothelial cells. We see that now when we compare the control mice to our vinculin cardiac specific knockout mice, that with loss of vinculin in the cardiac myocytes, we still have residual vinculin expressed again in non-myocyte populations. We see that there's, at that same time, loss of Zo1 from the vinculin knockout mice. And that's shown here in a blow up from that merge figure, where in the control litter mates, Zo1 and vinculin well co-localize at intercalated discs. But when vinculin's gone, likewise, Zo1 appears to disappear at these sites.
Bob, this is Rob. Steve Pulsing has got his hand up. I'm going to allow him to talk for a second.
Okay.
Where is he? Oh, man. Pulsing. I'm sorry.
No problem.
Okay, Steve. Dr. Pulsing, can you hear me?
I can. I'm sorry. It was an accidental hand raise.
Oh, all right.
All right. You're forgiven.
Thanks, Bob.
We'll move on. So to get further at this question, what we wanted to see were, does vinculin and Zo1 actually physically interact, or at least complex? And so we performed immunoprecipitations. And from this, we see that immunoprecipitations with vinculin brought down here in whole heart, and more so in isolated cardiac myocytes, Zo1. Likewise, the reverse was seen. And similarly, we could see that connexin 43 was also part of this complex. So from this, we could not say that there's absolute direct binding of vinculin to Zo1, but clearly that Zo1 and vinculin, and in fact connexin 43, appear to form a complex in cardiac myocyte extracts. So from this, we went ahead and with the help of Andy Dominighetti, that was at UCSD at the time and now is at the Rehabilitation Institute of Chicago, where he's studying skeletal muscle extensively, helped us with a 2-hybrid screen where we showed that vinculin indeed directly interacts with the third PDZ domain of Zo1, as shown here with this interactions, measuring beta-gal expression. Indeed, to get further at this, what we elected to do is to set up a neonatal mouse ventricular myocyte model. And to do that, we cultured the floxed homozygous vinculin cells, and then utilizing an adenovirus CRE could delete vinculin expression in the cell culture model. As we were concerned, as we've seen with many of the proteins that are so critical for cell matrix adhesion, if we fully deleted vinculin and other proteins from the myocytes, indeed many of the cells would not attach well or would not remain attached well to the dish. But we could reduce the vinculin expression significantly after about 36 hours. By 72 hours, the cells did not look very happy, and so many of our experiments occurred in that 48-hour to 60-hour window. And to get membrane expression of Zo1, we had to really have a packed dish of cells, as shown here, to get good expression of Zo1. Because our question was, if we then reduced vinculin in this cell culture model, would we basically see over the short term much of what we had seen in that mouse model? And here's the merge with alpha-actinin as a sarcomeric marker. And indeed, what we saw was that as vinculin was reduced, even modestly, here's about 70%, Zo1 went down, and a host of other proteins also now at the costamere, as well as at the intercalated disc, including talin, beta-1D integrin, which is a muscle-dominant isoform of beta-1 integrin, and connexin 43 were all reduced. And indeed, as we had shown that connexin 43 was in this complex with vinculin as well as Zo1, as we lost vinculin from the cells, here's the adeno-Cre infected dense cultures compared to a laxi control, connexin 43 was rapidly lost. We know how rapidly that turns over, whether it's in a tissue culture model or one in cell culture. And to study this functionally, we did both scrape loading and, as shown here, lucifer yellow dye injection. And in the control culture plates, we saw good spread in short time of the lucifer yellow dye here with DIC and lucifer yellow. But this was abated significantly in the cells that had lost or had reduced amounts of vinculin and Zo1 and of course, connexin 43. So from this work, both utilizing our mouse models as well as the cell culture models, we saw that in the mouse model, early on, they had preserved cardiac function despite significant loss of vinculin. I didn't show you all the data today, this is published, but that they developed sudden death. They had polymorphic ventricular tachycardia, altered intercalated disc structure, and abnormal connexin 43 distribution, despite the fact that it was still preserved in expression to a great extent. But later on, as the mice, if they escaped this period of sudden death, those that survived indeed developed a dilated cardiomyopathy that was quite profound and died quite early on. We then saw from the second part of this that vinculin co-localizes with Zo1 and connexin 43. As it's lost from the intercalated disc, Zo1 is likewise lost, as is connexin 43, which becomes both lost and lateralized, and that vinculin and Zo1 directly interact, as we saw from the co-IP and yeast 2 hybrid studies. And we posit that vinculin is therefore essential for normal connexin 43 positioning and function in the myocyte. And this occurs via this tripartite complex of Zo1, connexin 43, and vinculin. So this is how we conceptualize this, is that here's a normal myocyte, but if we lose vinculin or have a variant in vinculin, as shown here diagrammatically, then Zo1 disappears, and subsequently it destabilizes the entire intercalated disc, and we have loss of connexin 43. So with that in mind, and I'm happy to take questions now or at the end, what I'll spend the rest of the talk on, that's probably more of interest to many of you on this, but again, it feels like bringing coals to Newcastle, is to spend some time discussing this connected protein, zonulcludins 1, that we've studied for some time. And I'll mention that this project, and I believe he may have spoken at your institute some years ago when we could be there in person, Kirk Knowlton had started independently at UCSD. He elected to change pathways a bit. He's now at Intermountain Health in Utah, where he's pursuing more translational and clinical work. And since we had been studying vinculin, found this direct connection to Zo1, I kind of inherited this project, which was quite fortuitous from Kirk. And we've seen this come to fruition as we've included him throughout, despite him moving away from UCSD.
So Bob,
yes.
Here's the question. It's from a graduate student,
Sure.
William Adams, and he asks whether you looked at changes in RNA transcriptions for the transcription for the proteins that were reduced by vinculin knockout.
I'm sorry, Rob, did we look at the transcripts from the proteins?
Yes.
We did. They were reduced appropriately. I don't have all that. I didn't want to include all the data because I many times see PCR data in talks or in publications, and my question is usually the opposite, asking, well, that's fine, but how long lived is the protein, and is the protein reduced, which I think of always as the more functional aspect. Of course, we can think of many aspects of how transcripts can function beyond producing protein, but we did see parallel reduction of many of the transcripts, and I can try and dig that up at some point if this person's interested.
Thanks.
So moving on to Zo proteins in the heart, as I'm sure many or most of you on the call or on the Zoom are aware, there's 3 identified Zo proteins. Maybe you're even aware of others that I am not, Zo1, 2, and 3 that are encoded for by the tight junction protein genes. Zo1 and 2 are ubiquitously expressed, just as I was talking about with vinculin, talin, the beta integrins that we've studied. So this, of course, is somewhat frustrating and why we have to study it in a cardiac myocyte specific manner. And Zo3 is more restricted to epithelial cells. In the heart, the Zo proteins that are expressed in heart are found in the Adherens and Gap junctions in the intercalated disc, albeit our interest in them. As we're always interested in, before we undertake any sort of mouse modeling in a cardiac specific model, we want to know what happens in the global knockouts. And this was done many years ago, in that knocking out Zo1 globally leads to embryonic lethality about E11.5 in the 21-day gestation period. And these mice have defects in yolk sac angiogenesis and chorioallantoic fusion that leads to rapid demise in mid-gestation. The Zo2 global knockouts have also been created, and they are lethal very early on at gastrulation. And the Zo3 global knockouts, at least to my knowledge, has no obvious phenotype, but obviously it depends on how carefully one has looked. But that's what's in the literature. So some years ago, initially in Kirk's lab and in combination with others in our large group, we have about 9 PIs in our immediate conjoined laboratory that we really enjoy, that are very much aligned in many of their foci in cardiovascular medicine. We produced the Zo1 floxed allele, and again, used the alpha myosin heavy chain nuclear Cree driver to make our Zo1 cardiac specific knockout mice. And with that, here's a cardiac myocyte specific Western, because there are, as you'll see later on, there are differences if we do these sort of blots from ventricular tissue versus isolated cells. We could get really complete ablation of Zo1 expression protein from those myocytes. With that, we saw, as we had predicted, that Zo1, while it's still present at extracardiac sites, was completely lost from the intercalated disc. And here we're using desmoplakin as a marker of the intercalated disc, and here's the merge. And the only residual Zo1, and here again, as I showed you earlier on for some of our PCAM, for some of our vinculin studies, we used PCAM as a marker of endothelial cells, showing that the Zo1 that remained in ventricular myocardium was found co-localized with PCAM at endothelial cell sites. And so initially we were, of course, disappointed. There's one member of our group, no matter what knockout mouse they make, somehow within weeks after the mice are born, they have incredible findings or findings during birth. We've never been that lucky in most of our models. We had no myocardial histologic findings. We had normal basal contractile function. This is out to 90 weeks of age. These were very painful and expensive studies. There was no difference in control, and here, to be ultra fastidious, we used the heterozygous knockouts. Even when we performed, as we've done for decades, transverse aortic constriction to produce significant hemodynamic loading on the Zo1 mice. And we were really quite surprised, realizing that we had lost this protein from the intercalated disc. We expected, these mice to fall apart, as we've seen with many other analogous models that had important intercalated disc proteins. And so here's a demonstration of the histology. The picture is not perfect. There's some separation of myofibers. One interesting thing that we don't know fully how to read out is that in the Zo1 cardiac specific knockout, we actually saw increased thickness and increased normalized weights of bilateral atria, the left atria and the right atria here normalized to tibia length in our cardiac specific Zo1 knockouts compared to controls. What we did find, which was very compelling, is that Zo1 appears to have high-grade AV block. And although we really haven't cemented this identification, as I'll show in some parallel studies that were published simultaneously with a colleague, we saw loss of P waves, but we also saw significant, this was under mild isoflurane sedation, which we're always concerned, is it affecting heart rhythm? But with conscious telemetry, we saw different variations of high-grade atrioventricular block. We wanted to say we had complete heart block, but to be on the more conservative side of things, when we published this, we felt it was more traditional to think of this as really high-grade AV block. I think we need further studies to say that we have absolute complete heart block. And here we just show in a ladder diagram our Zo1 CKO mice that have normal production through to the AV node, and then there's block to getting to the ventricle as compared to the control mice shown here, where there's a P wave before each QRS complex, not so here. And here we did intracardiac electrograms with Yusu Gu, who's such a wizard in our mouse physiology lab, which again, with atrial pacing, here the pacing goes through on each of the beats, and here we lose that and could not pace within the Zo1 cardiac specific knockout. We did optical mapping. We have collaborated with our incredible bioengineering group at UCSD for many years, Andrew McCulloch and others in his group. And from this, we saw that there was AV delay as shown here, but that intrinsic ventricular activation the activation time was preserved, and that was shown in the optical maps, as I'll show here, and I'll show a video of that in a moment. Action potential duration was identical, and there was a trend towards intrinsic conduction velocity being slowed in the knockout mice as compared to control, as shown here. In left atrium, less so in right atrium, and not so really in ventricle. And here's from some of the videos that Andrew and his laboratory produced for us that shows with atrial pacing here at a cycle length of 150 milliseconds on this anterior view, we see consistent conduction in the control mice, but not so in the Zo1 knockout mice, in that pacing here in the atria does not always get down to the ventricle, and aligned with what we'd seen on the intracardiac mapping and the other studies. We've mated these mice into Sylvia Evans' HCN4 GFP knock-in mice, which have been shown by several groups. Obviously, there's a concern if one knocks into the HCN4 locus, which is present in the conduction system, and knocks out this hyperpolarization channel, whether one would affect conduction. And several groups around the globe now have proven that this is absolutely not so. We use this GFP marker simply to look structurally at SA node and at AV node as possible, and at least at the very gross observational level within myocardium in the control mice as compared to the knockouts, we saw no great distinction in the morphology of the SA node and AV node compared to controls, within the limitations of course, this type of assay. We looked at protein expression. This is now in heart tissue And we did this by chamber, realizing there may be differences, particularly when we saw that the atria had thickened walls and increased weights in RA and LA versus the right ventricle and left ventricle. Whether we looked here, we saw a reduction of Zo1, not so much of Zo2 at the whole tissue level. I'll show you at the myocyte level later. But again, at the whole tissue level, where we're dealing with myocytes, non-myocytes, et cetera, and conduction system cells, as well as ventricular myocytes, we saw no changes in vinculin, connexin 43, phosphorylated connexin 43, beta-catenin, connexin 30.2, cadherins, catenins, and connexin 40. And here at the immuno level, again, not so much difference in connexin 43 in the left ventricle. This goes along with the fact that conduction within the left ventricle was preserved, function was preserved, and this allied with the rest of the data that we had shown. But then we started looking at AV node, and with this, we saw that connexin 45 was reduced in our knockout mice in the AV node. This is shown here by Western blotting, here in the atria. And here in the AV node, we had nice staining. This is Jolin Zhang's work that he really championed the entire Zo1 project, and really it was quite a tour de force. And to isolate AV nodes, SA nodes, is really quite an undertaking in the mouse. So we localized this utilizing the HCN4 nuclear GFP mice that Sylvia Evans had loaned to us, my next door neighbor. We could then localize the AV nodal tissue. Here in the control mice, see nice expression of connexin 45, as shown here, now color-coded in red, with dominant loss in our knockout mice in the AV nodal region, which is so nicely marked by the HCN4 nuclear GFP. We saw that when we isolated myocytes now, a little more specifically in our Zo1 mice, that Zo2 became upregulated. And this is not AV nodal myocytes, but ventricular myocytes, as shown here. And here is AV nodal tissue, where we saw that Zo2, again, is present in the control mice, but is downregulated in that AV nodal tissue. Importantly, Kirk Knowlton's lab had published some years ago he has great interest in myocarditis and was interested in Coxsackie adenovirus receptor, or CAR proteins, with that in mind, that these CAR cardiac-specific knockout mice had conduction system abnormalities. And in fact, that's how he got interested in Zo1, really in a roundabout way from our interest with vinculin. And we saw that CAR protein was reduced in the atria in the Zo1 CKO mice, but not in a ventricle. And some of that staining is shown here. Here's right atria staining for CAR in red in the knockout compared to control. And here is the left ventricle, where it's still quite preserved. So this may be part of the process, but not fully explanatory of such. We saw that in the AV node here, not so much change in CAR protein, at least immunomicroscopically. And we became very much interested, and this is work we had to get help from Rob so nicely, who lent us the NAV 1.5 antibody. We know that his group and others, Mario Del Mar, and many around the globe have had great interest in NAV 1.5. And we wanted to see, would this be changed when we lost Zo1 in our mouse model? Indeed, aligned with no significant findings in the ventricle, we saw no significant change in NAV 1.5 expression in ventricular tissue. But when we looked at, here again, the HCN4 GFP marked AV node, we saw, at least in our hands, that NAV 1.5 was well expressed in AV node in the control mice, but was reduced, or for the most part lost, in the AV nodal tissue, but not so much in ventricle. And here's just a zoomed in version of that. Here's AV node marked by the blue-green HCN4-GFP. And here's NAV 1.5 staining, which was virtually significantly reduced and or lost in that AV nodal tissue. So in this, what we've reasoned out, and this absolutely requires more work, is that while we expected that with loss of Zo1 from the cardiac specific knockout, we would have abnormal contraction, contraction was preserved. Zo1 was lost in our knockout model compared to controls with alpha-myosin heavy chain Cre, but Zo2 remained in the ventricular myocytes, and as did NAV 1.5, the conduction was normal and contraction was normal, in that we feel that Zo2 can take the place of Zo1. Not so in AV node, where with Zo1 loss, there's not significant replacement or expression of Zo2, and we saw reduction of NAV 1.5 and connexin 45, leading to this high-grade AV block, including heart block. We think there's also SA nodal abnormalities, but really didn't include that because we didn't think the data was as strong as in our AV node. Now, I feel compelled to present this data in that as I was well along with this work, I called up a colleague that actually had been on study section with me, and perhaps with Rob, Ivan Moskowitz, to borrow, as we all do, various Cre mice. And Ivan said, well, I'm happy to send you this mouse model, but I know that you're now working on Zo1, and one of our junior investigators here at the University of Chicago, Chris Weber, who's a talented patch clamper and now trained MD PhD pathologist, is also working on this. So what do you think about you working directly with Chris? This was a couple of years ago, and we both were kind of struggling in getting our Zo1 projects going. And so I said, okay, well, this is a junior faculty member who's just getting his career going. I know where this, I've been in that same place. So we started a nice cooperative collaboration with Chris. He really did his own independent work, and we basically held our paper out of publication so that we could publish together in Circ Research. And Chris had a variation on the data that we presented, and I'll try and present that briefly to see how his work supports ours and vice versa. So he also created a floxed Zo1 model. This was completely independent of us, but he used an inducible form of Cre that we have used and others around the globe have used, a tamoxifen inducible mercury mirror model that releases Cre from being bound and then allows it to be targeted and flocks out alleles in an inducible manner from Jeff Mulkinton. And with that, Chris's lab showed significant Zo1 reduction, as we have for many of the proteins where we've used this. There's some residual. You get about a 70 to 75% reduction of Zo1, and that's shown here. And he, like we, showed here now slight variation, and this is now in adult mice. His Cre is only present after induction, after the mice have fully matured, and I think that's the variation in our model, in that he showed a prolonged PR interval, as he's showing here in his knockout mice compared to controls. And interestingly, he showed also complete heart block, where he's showing significant prolongation of the AH interval as opposed to the HV interval, with intracardiac mapping here in the knockout mice shown in red compared to the controlled mice shown in blue, with preservation of the HV interval. He showed that with loss of Zo1, connexin 40 was reduced. He suggested in his paper that CAR was reduced. I have to say it's not as dramatic as I'd like. I don't even know if it's statistically significant, but that connexin 40 was reduced along with Zo1. Being a pathologist, he had great access to human tissue and actually localized AV node in human tissue and showed how Zo1, shown here by staining an AV nodal tissue, shown here in brown. Co-localizes with connexin 40, as shown here. And interestingly, to his credit, he took this a step further and used, in addition to the cardiac myocyte specific Cree, now used a series of conduction system Crees that he obtained from my colleague Ivan Moskowitz that we had been interested in to begin with, an HCN4 Cree, just as we had marked cells. HCN4 is expressed throughout the conduction system. Chris used that to his advantage. And what he showed was with the HCN4 Cree, he's showing that diagram here in red, that it's distributed throughout the conduction system, that there was PR prolongation. And yet when he used more of an AV bundle Cree, KCNE1 Cree, there was no significant heart block. He also showed preserved ejection fraction, though tucked in some of the supplement in his paper in these inducible knockout mice. He showed that there was a slight decrement in left ventricular function. Again, it was quite slight. And here he showed, interestingly, that in his hands, he did not see a change in NAV 1.5 expression in AV node, albeit we looked in our mice who had Cree expressed for many weeks and months with loss of Zo1. He did this 10 days after tamoxifen injection and saw a variance here in his own hands. And so summing up Chris's work that I think, parallels ours and really is combining our work with his, really, it's always nice to see that what you're doing in the laboratory is supported by work independently by others at the same time, that with tamoxifen-induced loss of Zo1 in adult mice, we saw about a 70% reduction of Zo1 in his model. This leads to postnatal loss of connexin 40, perhaps a decrement in CAR expression, but 10 days after induction, no change in NAV 1.5 in his hands, but clear prolongation of PR as well as AV block with specifically prolonged AH intervals. And this was confirmed in his mice by use of that HCN4 Cree mice. To just show you where we're headed with this, and this is an unfunded project, as we all have buried in our labs, and hopefully we will be able to pursue this, but we absolutely have pushed this further along. We have Zo2 flox mice. In the Zo2 flox mice, we see increased Zo1, no change in Zo3, as we're always concerned about. This is in isolated myocytes, as shown here. And we've combined the Zo1 and Zo2 knockouts. And indeed, and this still is somewhat mysterious to us, by day 13.5, none of the double knockouts survive. Histologically, to date, when we look at these embryos, there's really no dramatic change. This is a couple of days before there's no survivors at E11.5. Minimal evidence of increased thickness of the compact layer, perhaps here in the knockout, the double knockout compared to controls. Perhaps some blebbing of the pericardium. One always questions this. We think from early days that these actually have reduced heart rates, and perhaps that's the etiology from their demise. A lot more work to do on this double knockout, but quite interesting. No obvious morphologic changes. And I'll wind up with, the question that always comes from so much of my work. I have so many colleagues who are basic scientists, have no clinical background, and I always see doing many times more translational work than I do. And many people always say, you're working on these very basic, ubiquitously expressed proteins. Is there really any clinical relevance? So I feel compelled to talk about some of this. There is some, but it's not really found in the general population, as if we had variations of many of the proteins we study, unfortunately, neither mouse nor people may survive. But there are variations in these proteins. Mike Ackerman has so championed so much of the genetics of cardiomyopathy and published now many years ago, this was 15 years ago, variations in this is metavinculin, that muscle-specific variant of vinculin being associated in families with hypertrophic as well as dilated cardiomyopathy. There have been other vinculin variants, this is more recently, that have been identified in families with sudden unexplained nocturnal death syndrome with decreased cardiac sodium currents, allied to some of the work that we've alluded to from the Zo1 model as well as the vinculin model. And in really a tour de force study produced by Decker Deacon, who's now a dermatologist, Rob, take notice, he's at Utah. He's a very highly trained MD PhD geneticist, and he was a MSTP student in Neil Chee's lab. Neil and Decker really championed the genetic work in this. This was really a fabulous collaborative effort that 3 groups really fully partake in. My group worked on the mouse work, and Adam Engler, one of our bioengineers, worked on the stem cells, where we showed that a variation in vinculin in combination with a tropomyosin vinculin led to a dilated cardiomyopathy and actually, unfortunately, sudden death in a family that had these variants. So vinculin variants are present in human disease. They're pretty rare. If one does panel selection as opposed to a whole exome or whole genome surveys of individuals with possible genetic variations of dilated cardiomyopathy, vinculin is on those panels. What about Zo1? Less data there. This came out a couple of years ago. I'm sure all of you that are Zo1 experts are attuned to this, that in a whole exome sequencing study, they found variations in Zo1 that might lead to arrhythmogenic cardiomyopathy. There were highly preserved variants across species, as shown here. And they showed that a couple of these variants in German and Italian families might lead to arrhythmogenic cardiomyopathies, and one of them might lead to both dilated cardiomyopathy and, interestingly, ventricular tachycardia. They went through, and this is part of the struggle we have if we think about genetics in the clinic, when we look at variants and have to explain a variant to a patient, there's many different ways and that these various programs evaluate whether these variations in a sequence and in a protein ultimately will be damaging, disease-causing, neutral. And we can see that as we look at these various Zo1 variants I just mentioned, there's variation in a theme depending upon which program one's used to explain how the variation might affect causality of disease. And then if we look at how predictions might be made of what these variations might do to Zo protein itself, there's some that have decreased stability and others that actually have increased stability. So we're in a conundrum of how these may function in leading to human disease. And I think it's very early days, but I think it's very clear that these proteins that we're all interested in the intercalated disc may well lead to disease. So here I'm just winding up, showing a model that was published along with our Circ Research data, showing how loss of Zo1 might affect intercalated disc function. And of course, I need to thank everyone that's involved. This is the building that we're in at UCSD, a view from UCSD out towards the beach there. The Vinculin project was championed by Elise Zemlich-Harp, who was in the lab, who's still at UCSD. Alexander Petoshin helped with some of the scrape loading and injection studies, and Anna Dominichetti with some of the yeast 2 hybrid. As I mentioned, Kirk's lab started the Zo1 project. He's now at Intermountain Health. Jolin Zhang, who started with him, continued with me, and is still at UCSD, really almost single-handedly did most of the work on Zo1, along with Kevin Vincent, a graduate student in Andrew McCulloch's lab, and Angela Peter, that's at the University of Colorado. Other members of the laboratory that are working on different projects, tremendous amount of collaborators at UCSD, some of which I've mentioned earlier in the talk. Our long-term collaborator, Josh Goldhaber at Cedars-Sinai, and Rob that helped us with the NAV 1.5. Our funding from NIH, from the Veterans Administration. We recently have gotten a California Tobacco Research Program grant on a completely different topic. And hopefully I'll come back. And this was early on when I was supposed to come early on in the pandemic, and I was getting ready and pretty nervous about flying out. And Rob called one morning and said, "You're not coming. We've just closed the institution." And at that time, I saw this, that maybe I'll be able to come and bring some of these chocolates, and that'll be the only evidence of COVID that we'll have. So I'll end there and stop sharing my screen, and I'm happy to answer questions.
Yeah, that's awesome. Awesome talk, Bob. And we do have some questions. The first one's from Steve Pulsing, and I'm actually going to make Steve live because...
Is it a mistake or is it real?
He had a question from the earlier part of the talk, but maybe listening to the rest of your talk, he has got some further questions. So, Steve, can you hear us?
I can. This is not a mistake this time. Thank you, Bob.
Okay.
Great talk. Really enjoyed that. I had a question about the vinculin knockout mouse. So if torsade de pointes is often preceded by a bradycardia before you get that short, long, short, did you notice any QT prolongation or bradycardia in the vinculin? I know you went over it in the subsequent knockout mouse.
Well, at least you're asking me about whole EKGs as opposed to patch clamping, which you have much more expert in than I am. You know, even in people, when clinical studies are done, QT prolongation is something that the computers aren't even good at, and people pay our cardiology fellows and or clinicians to tell when they're assessing drugs, for instance, as having bad side effects, is the QT prolonged or not? To do that in the mouse, you know, the EKGs in the mice are quite different than human. I think there are, you know, people that publish about QT prolongation. Perhaps you have, and I'm offending you by this response, but I think to get at measuring QTs in mice and to say it's absolutely prolonged is not a simple task. So the bottom line answer is I don't have an answer for you, and I think it's very difficult to get at. With intracardiac electrograms, I think we can get more at that. We don't have that, and I don't recall whether there was slowing before there was the you know, the VT. I mean, interestingly, as you probably well know, mice are incredibly resilient in that they can have prolonged episodes of ventricular tachycardia and still recover.
Right.
Yet these mice ultimately succumb to this. So we'd have to go back to our recordings, and we could do that if you're interested.
I am. Thank you again. That was great.
All right. Next question is from Anthony Lamantia. Anthony is a professor at the Institute, the director of one of our neurosciences centers, and I'm going to allow Anthony to talk so he can ask the question himself. Go ahead, Anthony. Anthony, can you hear us? You're still on mute, I think. There you go.
There we go.
Rob, why don't you just read the question to him?
Okay. Yeah. Seeing as Anthony's not coming through, so he asked, since disrupting junctional connections between cardiac myocytes is likely to elicit multiple downstream changes, how direct are the effects on additional junctional proteins? Is the loss of genes like Zo1? How dramatically is the overall transcriptional landscape in the cardiac myocytes change? So I guess there's two questions there.
If I'm reading into this, I guess the question would be, have we done RNA-seq, have we done proteomics, et cetera, on these models? And for the Zo1, we have a little data, nothing that I can tell you about. The question always is from some of these studies is when to do this. In other words, I think in the Zo1, this is definitely tenable in that there's an absolute window where we don't see conduction abnormalities after we knock it out. And particularly if you use, you know, for instance, Chris Weber's inducible model, that one can then isolate tissue or isolate cells and look at changes in transcripts and or proteins before there's any functional effects. There's always the question of, you know, you're just not looking hard enough at the functional effects. So the bottom line is I don't have data for this. We're very much interested in that. You know, do we go to the level of doing single cell RNA-seq? We don't have any of that data. And I absolutely would be, I agree with you. We've shown you that a host of proteins can be affected with ablation of just one of these, and that it destabilizes, whether we're talking about intercalated disc junctions or costameric junctions. And absolutely, this is probably a combinatorial effect, not just the single loss of the gene, but that there's destabilization and loss of multiple proteins at the same time that leads to these phenotypes. So I don't know if I answered your question.
Okay, next question is from Spencer Marsh. Spencer's a postdoctoral fellow at the Institute, in fact, in my lab, and I've unmuted Spencer. So Spencer, I'll give you a chance to ask your question yourself. If I can't hear you, I'll go ahead and ask your question for you.
Awesome. You can hear me?
Yep.
Sweet. All right. Thank you, Dr. Ross. That was really great. You showed pretty conclusively that Zo1 has a huge role in cardiac conduction in cardiomyocytes. Have you done anything in like a post-infarct model or an injured model in which myofibroblasts retake that role from cardiomyocytes? And if the kind of overall lack of function in myofibroblasts is possibly due to a lack in Zo1?
So we'd be very much interested in that. We actually wrote some of this information in our unfunded NIH grant, but we haven't done those studies. We are studying the role of many of these proteins in post-infarct remodeling, ischemia reperfusion, but we haven't done it in the Zo1 model. And I agree, it could be quite interesting to do that. Obviously, to target fibroblasts, we've done some of, we initiated some of these studies, not so much with Zo1, but with other proteins that we've studied that are ubiquitously expressed, and we've knocked them out in myocytes. We'd like to do that in fibroblasts. Obviously, things like the periostin Cree and things of this nature didn't get very far with some of the proteins we were interested in, and I think it really depends on the model. But to look at it specifically in the myofibroblasts, maybe we could do it in a cell culture model, but those are, difficult studies, but a great question.
Thank you.
And we have a question from Xiaobo Wu, and Xiaobo is a graduate student in our Translational Medicine, Biology and Health program, and I've allowed him to ask his question in person. Xiaobo, can you hear me? Can you unmute and ask your question?
There he is.
Yeah. Hi. Thank you, Dr. Ross. I have a question about so you have a knockout of Zo1, so that causes no loss of negative 1.5. Did you do an experiment to figure out if is there a functional change of negative 1.5?
So this is opposite, so I showed you, we saw changes in AV node. Chris Weber, but in a different model, and 10 days after loss of Zo1 did not. Absolutely, we need to do what you're asking. And this is what, my friend and colleague at Cedars-Sinai, Josh Goldhaber, is actually, a bona fide patch clamper, does fast confocal, and is specifically interested in recent years in SA node and AV node. And these are the exact studies that we have planned to do with him. I will tell you, we'd hope we'd have these done by now, and COVID has absolutely slowed things down in my lab. And when COVID was shutting things down, we really shrunk our Zo1 colony, and to get it back up, we have to do that. So we absolutely want to do that. And whether it's just NAV 1.5 or there are other channels affected, that's our goal.
Thank you.
Okay, thanks, Salvo. I should have mentioned that Salvo is in Steve Polsing's lab. We have a question from another graduate student, Randy Strauss, who is in my lab, and I'm going to unmute Randy and allow him to ask his question in person. Randy, can you unmute yourself and address Dr. Ross?
Yeah, can you hear me?
Yep.
Oh, cool. Yeah, so you showed that Zo1 is dominantly expressed in the endothelia within the heart. So I was wondering, what your thoughts are on the role the vascular plays in mediating the alteration of cardiac function in Zo1 knockout models.
Well, in the global knockout, I think that is clearly role. In the models that I used in my lab and Chris Weber used in his lab, we really know that those Cre proteins are pretty well restricted to muscle. But we absolutely agree with you that we have a great interest in that. And actually a colleague on our campus that's interested more so in cavernous venous malformations in the brain has taken our mice and is looking at some of that. We did, or Jilin Zhang with Kirk, one gets your Flox model, and once it's functioning properly, the ideal thing to do if you had unlimited funds is to take a bunch of Cres that are specific to various cell types and to do this. And I know that the Ty2 Cre Z1 model was made, and yet there was a phenotype. We never really came to fruition on that because there's only so many mice one can maintain in a colony and people that are available to the studies. But I'm guaranteeing that, as you're alluding to, there's a significant role of Z1 in the endothelium. And
Great.
we'll find out hopefully soon.
Great, thanks a lot.
All right, Bob, that was our final question. So it's just up to me to thank you once again for an awesome seminar. Really enjoyed it, and great work.
Thank
Mike.
you for inviting me. Thank you. So I